Right now, like inside almost every single one of your cells, there is this literal microscopic hourglass.
Yeah, it really is a physical thing.
Right. And with every breath you take, or every time your heart beats, every time your skin cells replace themselves, a few tiny, invisible grains of genetic sand just fall away.
They just drop right to the bottom.
Exactly. We've spent centuries talking about the biological ticking clock as if it were just a metaphor, right? Like some abstract shadow hanging over us.
It's an incredibly pervasive cultural idea, but the reality is actually much more profound.
Yeah.
Yeah, I mean, you don't have to rely on poetry to describe aging. You can literally put the biological clock under an electron microscope and just, watch it tick.
And that is exactly what we are unpacking today. Welcome to the deep dive. Whether you're a clinician trying to navigate new oncology drugs, or someone in the biotech business tracking the longevity market, or just someone listening on your commute who wants to understand what aging actually is at a physical level.
Right, we are looking at some incredibly dense molecular biology today.
We are. We've got genetic sequencing data, FDA clinical trial pipelines, translational gerontology studies, and the mission of this deep dive is to understand this ultimate biological countdown timer,
The telomeres.
the telomeres, and the enzyme that actually holds the power to rebuild them, which is telomerase.
Yeah.
We want to separate the hype from the hard science.
Because it really is the highest stakes game in modern biology. I mean, manipulating the system is the holy grail.
The ultimate prize being cellular immortality, basically.
Exactly. But what the hard science shows us and what makes this so intensely complex is that the exact same molecular mechanism that keeps a cell young and vibrant, it's the identical mechanism that makes a cancer cell virtually unstoppable.
It's the ultimate double-edged sword. So, we need to understand how the machine actually works before we can talk about trying to hack it.
Definitely. Let's start with the anatomy.
Okay, so in the literature, there's this really brilliant visual analogy that comes up repeatedly when describing telomeres. They're often compared to aglets.
The aglets, yes. It's a classic analogy for a very good reason.
And for those who aren't familiar, aglets are those little plastic or metal caps on the ends of your shoelaces, right? The ones that keep the strings from fraying into a totally chaotic mess.
Right. And to understand why your DNA actually needs an aglet, you have to visualize the architecture of the human genome.
Okay, paint a picture for us.
Well, unlike bacteria, which generally have circular DNA, so like a continuous loop with no beginning and no end, human chromosomes are linear.
Linear, so they're just long, straight, fragile threads of genetic information.
Exactly. And having two distinct ends floating around in the nucleus presents this massive vulnerability.
Like a catastrophic vulnerability, according to the source.
Right, absolutely catastrophic. You have to think about this from the perspective of the cell's internal security system.
Right.
Our cells are equipped with highly aggressive DNA repair machinery, and its entire job is to patrol the nucleus, just looking for broken DNA strands.
Because normally broken DNA means something terrible has happened, right?
Right, like radiation or chemicals snapped a chromosome in half. When that happens, the cell's emergency systems absolutely panic.
They just freak out.
Total freak out. They grab those two broken ends and desperately try to glue them back together to save the genetic code.
Okay, let's unpack this a bit. Because if I'm understanding the architecture correctly, a natural, healthy human chromosome is basically just one giant broken strand, right?
Essentially, yes.
It just stops.
Yeah.
So how does the cell know the difference between a natural ending and a catastrophic break that needs fixing?
And that is the brilliant function of the telomere. A naked, unprotected end of a chromosome looks identical to a severed strand.
So the cell would just try to glue everything together.
Exactly. If the chromosome just ended abruptly, the cell would grab the end of chromosome 1 and aggressively fuse it to the end of chromosome 2.
Oh, wow.
Yeah, you'd get these massive, tangled, mutant chromosomes that would literally tear the cell apart the next time it tried to divide.
So the telomere acts as a sort of biological disguise.
It does. It hides the raw end of the data.
So what is the disguise actually made of? It's not a different material, right? It's still just DNA.
It is still DNA, yeah, but it is entirely non-coding. It contains 0 instructions for building proteins.
Oh, interesting. So it's just feller.
It's a highly repetitive structural sequence of nucleotide bases. In humans, and actually across all vertebrates, that exact sequence is TTDGG.
TTG.
Right. Thymine, thymine, adenine, guanine, guanine, guanine.
So just 6 letters.
Just a 6-letter word. But it is repeated over and over and over again, literally thousands of times.
Wow. So it's creating millions of base pairs that form this massive physical buffer zone at the very tips of the chromosome.
Exactly. But that buffer zone isn't permanent.
Right. And the sources really highlight this deeply rooted mechanical glitch in our biology. It's referred to as the end replication problem.
Yes, the end replication
This
process.
is where the clock actually starts ticking, right?
It is. It's a fundamental, inescapable mechanical limitation in how our DNA copies itself.
Okay, how does that work?
Well, when a cell divides, it has to replicate its entire 3 billion letter genome, so the new daughter cell gets a complete copy, right?
Right.
The molecular machine tasked with this heavy lifting is an enzyme called DNA polymerase.
DNA polymerase. I always picture that like a train speeding down a track, reading the old DNA and building the new strand right behind it.
That's actually a really good visualization, but it's a train with some very specific, highly restrictive rules.
Okay, what kind of rules?
First, it can only travel in one single direction along the DNA strand.
Okay, one-way track.
Exactly. And second, and this is the really crucial part, it cannot just start building a new track out of thin air. It requires a starting block.
What kind of starting block?
It needs a short, temporary stretch of RNA, which is called an RNA primer, to be laid down first.
Okay, so someone else has to start the work for it.
Right. An entirely different enzyme comes in, lays down this little RNA primer on the DNA, and then the DNA polymerase train attaches to that primer and starts chugging along, copying the DNA.
Okay, I see where this is going. If it needs a primer to start, what happens when it reaches the absolute edge of a linear chromosome?
You've just described the exact mechanical glitch. The train reaches the end of the line.
And there's no space left.
Exactly. The polymerase gets to the very distal tip of the telomere, but there's no more physical real estate left to place that necessary RNA primer.
The train literally runs out of track.
It runs out of track. It just falls off before it can copy the very last segment of the DNA.
So the very tip is just left uncopied, which means every single time the cell divides, the new copy is biologically obligated to be a little bit shorter than the original.
Exactly. It's unavoidable. This is actually the marginotomy theory, originally proposed back in the 1970s by Alexei Lovnikov.
He was a Russian theoretical biologist, right?
Yes, and it was simultaneously observed in viral DNA by James Watson. Lovnikov brilliantly deduced that because of this mechanical limitation, the telomere must lose a small chunk, maybe 50 to 100 base pairs, with every single cell division.
So the shoelace cap essentially frays, like a little piece snaps off.
And this happens in virtually every dividing somatic cell in your body every single day of your life.
Wow. Here's where it gets really interesting, though. The sources get incredibly detailed about the physical structure of this cap. It isn't just a raw, blunt sequence of TTG hanging out in the cellular void waiting to get shipped away.
No, not at all.
There is a massive, highly sophisticated security detail surrounding it.
You can't just have exposed DNA floating in the nucleus, even if it's non-coding.
Because the alarms would still go off.
Exactly. The repetitive DNA sequence is just the foundational scaffolding. The actual protective function is carried out by a massive specialized protein complex known as shelterin.
Shelterin. I mean, it literally shelters the DNA. It's a great name.
Biologists sometimes get naming right.
Just sometimes, yeah.
But shelterin really is an architectural marvel. It's composed of 6 core proteins, and the literature heavily highlights 3 major players.
TRF1, TRF2, and POT1, right?
Yes. And these proteins don't just randomly stick to the DNA. They bind exclusively to that specific TTGG sequence.
Okay, so what do they actually do? Are they just forming like a hard shell around the end of the chromosome?
It's way more dynamic than just a static shell. They physically manipulate the shape of the chromosome.
Manipulate the shape. How?
To understand this, we have to look really closely at the very tip of the telomere. It's not a clean, blunt cut where the 2 strands of the DNA double helix end perfectly flush with each other.
Like a cleanly cut piece of rope?
Right, it's not like that at all. One of the strands, specifically the strand rich in guanine bases, is actually longer than its partner.
Oh, really?
Yeah, it extends further out, creating what biologists call a single-stranded 3' overhang.
So there's just a loose single thread dangling at the very end of the aglet.
Precisely. And in the world of molecular biology, a loose, single-stranded piece of DNA is a massive red flag.
Right, because it looks like a broken strand.
Exactly. It is highly unstable, and it just screams DNA damage to the cell. So the sheltering complex has to hide it.
How does it hide it?
A protein called POT1, which actually stands for protection of telomeres 1, specifically latches onto that single-stranded overhang to cover it up.
It acts like tape over a loose wire.
Yes, but the system goes a step further to ensure total security. Another shelter-in protein, TRF2, grabs that entire single-stranded overhang and physically bends it backward.
It bends it back.
It holds it back onto the main body of the telomere.
Wait, it folds the DNA back on itself.
It doesn't just fold it, it forces it to invade the double-stranded DNA further up the line.
What do you mean invade?
The complex physically pries apart the double helix of the chromosome and tucks that loose single-stranded end deep inside, where it base pairs with the complementary strand.
That is wild. It's literally tying itself into a knot to hide the loose end.
It creates a highly secure three-dimensional structure, and under an electron microscope, you can actually see this, it forms a large loop known as a T-loop, or telomere loop.
Okay, a T-loop.
And at the exact spot where the strands were pried apart to let the end tuck in, you get a smaller displacement loop called a D-loop.
Okay, let's unpack this.
Yeah.
If I'm visualizing this, the sheltering complex and these elaborate T-loops and D-loops, they essentially act as a molecular witness protection program.
That is a brilliant way to frame it.
Like they hide the raw ends, they tuck the loose threads away, and they completely mask the identity of the chromosome's end. So the cell's internal alarm systems are none the wiser.
Yes, exactly. Because the cell has two major alarm pathways, governed by proteins called ATM and ATR.
And the knot keeps them quiet.
TRF2 hides the T-loop structure so the ATM alarm doesn't go off, and POT1 cuts the loose thread so the ATR alarm stays silent. It's a perfectly balanced security system.
But it's a security system with a built-in expiration date, right? Because of that end replication problem, the telomere is still getting shorter every time the cell divides. The track is shrinking.
And that shrinking has severe structural consequences. As the telomere gets shorter and shorter, there is simply less physical space for those TRF1 and TRF2 proteins to bind.
Right.
Eventually, the telomere becomes too short to fold back on itself. The T-loop cannot form.
The knot comes undone.
The knot comes undone, the witness protection program fails, and that raw, single-stranded DNA is suddenly exposed to the nucleus.
And the alarms go off.
Deafeningly. But we need to pause here because there is an obvious paradox here that biologists wrestled with for a long time.
Okay, what's the paradox?
If our DNA inherently shortens and frays every single time a cell divides, how is it mathematically possible that a human being grew from a single, microscopic fertilized egg into an adult with roughly 37 trillion cells?
Right. If the track gets shorter every time, we should have run out of telomeres entirely before we even made it out of the womb. The math just doesn't add up.
The math only works if there's a way to put sand back into the top of the hourglass.
There has to be a biological countermeasure.
And there is. It's an enzyme called telomerase.
Telomerase. Okay, if telomeres are the physical ticking clock, telomerase is the invisible hand that can literally reach in and rewind the hands of time.
Exactly.
Let's really dig into this because this enzyme seems like magic. What exactly is it and how does it rebuild the shoelace?
Well, it's not magic, but it is an astonishing piece of biological engineering. Telomerase is a ribonucleoprotein complex.
Ribonucleoprotein, meaning it's a hybrid, part RNA, part protein.
Exactly. It operates as a highly specialized reverse transcriptase. Most people know transcription as the process where DNA is read to make RNA.
Right.
Telomerase does the exact reverse. It uses an RNA template to physically write new DNA.
It writes DNA from scratch.
It consists of two massive distinct components that work in tandem. First, you have the catalytic engine. This is a protein called TERT.
TERT, which stands for telomerase reverse transcriptase.
Yes. TERT is the physical machinery that grabs nucleotides floating in the cell and links them together. But a machine is useless without a blueprint. It needs to know what sequence to build.
And it needs to build TTAG.
Right. And that is where the second component comes in. It's called TERK, the telomerase RNA component.
Okay. So
TERK is a long, intricately folded piece of RNA, and embedded right in the middle of it is a tiny sequence that is the exact complementary mirror image of our human telomere.
So TERK is the instruction manual.
It is the template. When a telomerase reaches a shortened chromosome, the TERT engine grabs the end of the DNA, aligns it with the TERK RNA template, and starts manufacturing brand new telomeric repeats.
So it reads the blueprint, grabs a thymine, another thymine, an adenine, and 3 guanines, and stitches them directly onto the fraying end.
It physically rebuilds the track.
That is just incredible.
Yeah.
But reading through the research, this process isn't just Turt and Turk meeting up for a coffee and building some DNA. There's an entire wildly complex cellular logistics network required just to get this machine assembled.
Oh, absolutely.
The sources list a whole entourage of accessory proteins.
The assembly pipeline for telomerase rivals anything in a modern automobile factory. You can't just synthesize Turt and Turk and expect them to function.
Why not?
RNA, by its very nature, is highly unstable in the cell. If Turk were just floating around naked, cellular enzymes called ribonucleases would shred it to pieces in seconds.
So it needs bodyguards.
It requires a highly specialized security detail. The sources detail 4 essential accessory proteins. They are dysturin, NOP10, NHP2, and GA1.
They genuinely sound like a squad of droids from Star Wars.
They absolutely do, and their job is critical. These 4 proteins physically bind to the Turk RNA, forming a protective scaffold around it.
So they keep it from getting shredded.
They stabilize the complex and ensure the blueprint survives long enough to be used. But that's just the RNA side. The protein engine, TERT, has its own logistical nightmare.
What happens with TERT?
TERT is synthesized out in the cytoplasm of the cell, outside the nucleus. It's a massive, complex protein, and it's very prone to misfolding.
So it needs help getting into shape.
Right, so the cell recruits molecular chaperones, specifically HSP90 and P23.
Chaperones. I love that.
These chaperones grab the newly minted TERT protein, help it fold into its correct 3-dimensional shape, and literally escort it through the nuclear pores into the nucleus.
So you have the TERT blueprint being guarded by the droid squad, and the TERT engine being escorted by chaperones, all meeting up in the nucleus.
And even then, they don't assemble themselves. 2 additional helper proteins, reptin and pontin, act as mechanics to finally fuse the engine and the blueprint together.
Okay, so the factory's assembled. Is it ready to build?
Not yet. Once fully assembled, a protein called TCCB grabs the entire telomerase complex and physically drags it into specialized compartments within the nucleus called Kajal bodies.
Kaggle bodies. Are those like storage lockers?
More like specialized staging areas or maturation plants. Inside the Kajal bodies, the telomerase complex undergoes final processing and is held in reserve until the exact moment the cell divides.
And only then is it deployed.
Only then is it deployed to the telomeres to actually do its job.
The sheer complexity of that. Discarin, chaperones, reptin, pontin, Kajal bodies. I mean, all of this energy, all of this cellular infrastructure, just to put 6 tiny letters, TTAGG, back onto the end of a chromosome. It shows how profoundly vital this process is.
It is the very definition of essential. Without it, complex multicellular life simply could not exist.
But this leads to the most glaring, frustrating paradox in human biology, right?
Yeah.
Because if this telomerase factory exists, if our bodies have the exact genetic blueprints to build this time-reversing enzyme, and we know exactly how to assemble it,
Well,
why do I have gray hair? Why do our joints ache? Where is this enzyme actually working in my body right now?
that question is the absolute bedrock of modern aging research, and the answer is both brilliant and deeply tragic.
Okay, tell me the tragic part.
When you are a developing embryo growing in the womb, your cells are dividing at a furious exponential rate. To support that growth, telomerase is running at full capacity, 204 to 7. It keeps your telomeres long and pristine.
Building the house.
Exactly. But as we transition from embryos into fully formed, mature humans, our biology executes an incredibly deliberate, systemic shutdown.
By shutdown.
In the vast majority of our somatic cells, meaning our regular body cells, our skin, our muscle, our brain tissue, the body completely locks down the transcription of the TERT gene.
It turns the factory off.
It puts a permanent padlock on the engine room.
Oh.
Interestingly, the blueprint, the TERT RNA, is still floating around in many tissues, being guarded by those accessory proteins.
But without the TERT engine to do the actual building, the blueprint is totally useless. The assembly line is dead.
Completely dead.
So just to be absolutely clear, we possess the literal fountain of youth in our DNA, and our own bodies actively choose to turn it off.
What's fascinating here is how absolute that somatic lockdown is, but also how surgical the body is about granting exceptions, because the body isn't stupid.
Right.
There are certain cell populations that simply must continue to divide rapidly throughout your entire life. If telomerase were turned off everywhere, you would die in a matter of weeks.
Okay, let's explore those exceptions. Where is the padlock left open? Who gets to keep the key?
The most obvious exception is adult stem cells. Think about your gut. The epithelial lining of your intestines is subjected to massive physical and chemical wear and tear.
It has to completely replace itself every few days, right?
Exactly. The stem cells living deep in the intestinal crypts are constantly dividing. To support that, they are allowed to keep a low, persistent level of telomerase activity.
That makes total sense. If they didn't, your gut lining would just disintegrate. What about other organs?
The liver is a great example of dynamic control. Hepatocytes, or liver cells, generally sit quietly without much telomerase.
But they can regenerate.
Yes. If you suffer acute liver damage, say from a toxin or partial surgical removal, the liver cells can transiently reactivate the TERT promoter. They turn the factory back on just long enough to fuel an explosion of cell division to rebuild the organ, and then they shut it off again.
That is incredibly smart. It's like an emergency backup generator. What about the immune system? We are fighting off bugs constantly.
The immune system is entirely dependent on telomerase. When your body detects a new pathogen, a single specific B cell or T cell that recognizes that exact virus has to clone itself millions of times over the course of a few days.
To mount an effective defense.
Right. This is called clonal expansion.
Millions of divisions in a few days would obliterate a telomere.
It would wipe it out completely. Without telomerase, the immune cell would hit the Hayflick limit in the middle of fighting a simple cold.
And the immune response would just collapse.
And you would die from a minor infection. So activated lymphocytes are granted the ability to dramatically upregulate telomerase activity during an infection.
And I assume reproduction has to be an exception, right?
Yeah.
If I have a child, I'm passing my DNA to them.
Yes, the male germ cells, the spermatogonia that produce sperm, they maintain robust, constant telomerase activity. This is arguably the most vital exception for the survival of the human race.
It ensures that the genetic material packaged into a sperm cell starts with completely fresh, maximum-length telomeres.
Because if it didn't, a 40-year-old father would pass on 40-year-old shortened telomeres to his baby.
Exactly. And then that baby would pass even shorter telomeres to their child.
Without telomerase in germ cells, every successive generation of humans would be born biologically older than the last, and our species would literally age itself into total extinction within a few generations.
Okay, so the body perfectly regulates this. It gives the enzyme to the immune system to fight disease, the stem cells to repair critical tissue, and the germ cells to ensure the species survives.
Right.
But for the rest of us, for the skin on my face, for the cartilage in my knees, for the neurons in my brain, the clock is strictly ticking. The factory is closed.
It is a strict mathematical limit. By turning off telomerase in your somatic cells, your biology sets a hard, inescapable ceiling on exactly how many times those cells can divide before they run out of track.
Which brings us directly into the consequences of running out of track. Let's talk about what actually happens when that limit is reached. The sources refer to this ceiling as the Hayflick limit.
This is a foundational concept in biology. It was named after Dr. Leonard Hayflick. Back in the early 1960s, the prevailing dogma in science, largely championed by a guy named Alexis Carrel, was that all human cells grown in a lab culture were inherently immortal.
Wait, scientists genuinely believed that?
They did. They believed that if you just gave cells enough nutrients, they would divide forever.
But Hayflick proved them wrong.
He shattered that dogma. Hayflick was cultivating human fetal fibroblasts, which are connective tissue cells. He noticed that no matter how perfectly he maintained the culture, how perfectly he fed them, the cells would divide vigorously for a while, and then they would start to slow down.
They hit a wall.
Eventually, after roughly 40 to 60 divisions, they would just stop. They hit an invisible wall and refused to divide again.
And for decades after that experiment, nobody knew exactly what that invisible wall was.
Right.
They just knew the cells were counting divisions somehow.
It took years for the molecular biology to catch up, but now we know exactly what the Hayflick limit is. It's the shoelaces.
It is the end replication problem steadily chipping away at the telomere until the physical structure collapses.
Exactly.
So let's walk through that collapse. A cell divides 50 times. The telomere gets shorter and shorter. Finally, it reaches a critical length. What happens?
We go back to the sheltering complex. The telomere becomes so truncated that there simply isn't enough TTG sequence left for TRF1, TRF2, and POT1 to bind.
There's not enough room.
Right. So the protective T-lip cannot physically form. The knot unties.
And the raw, single-stranded tip of the chromosome is suddenly laid bare in the nucleus.
Yes. And the moment that naked DNA is exposed, the cell's internal DNA damage response, the DDR, goes absolutely haywire.
The alarms.
The ATR and ATM alarm pathways detect what they think is a catastrophic double-strand break. They immediately recruit powerful tumor suppressor proteins, specifically pathways involving p53 and p16RB.
These proteins act like emergency brakes.
They slam on the emergency brakes of the cell cycle. They flood the nucleus with signals that fundamentally alter the cell's programming. The directive is absolute. Your genome is unstable. You are a danger to the organism. Do not divide again.
So the cell stops dividing. Does it just gracefully die and clear out of the way?
This is where the story of aging gets incredibly grim. It doesn't die.
It doesn't.
No. If the DNA damage were caused by massive radiation or chemical spill, the p53 pathway would trigger apoptosis, which is a clean, organized, programmed cell death. The cell packages itself up and is consumed by macrophages.
A noble sacrifice for the good of the tissue.
Exactly. But critically short telomeres often don't trigger apoptosis. They trigger a unique alternative state called cellular senescence.
The zombie cells.
Zombie cells is the perfect scientifically accurate shorthand. These cells are permanently arrested. They will never ever divide again. But they are metabolically viable.
Meaning they are alive.
They are alive, they are consuming glucose, they are producing energy, and they just sit there in the tissue. That honestly doesn't sound that bad. I mean, so what if a few skin cells or joint cells stop dividing? It just means the tissue stops growing. Why does that make me feel old? Why does it cause wrinkles or arthritis?
Because senescent cells are not sleeping quietly. They are angry. They are terrified.
Terrified.
When a cell enters senescence due to telomere dysfunction, it undergoes a massive epigenetic shift. Its entire gene expression profile changes. It morphs from a functional, structural cell into a toxic factory.
A toxic factory. What is it making?
The sources refer to this phenomenon as the SASP, the senescence-associated secretory phenotype.
The SASP. Okay, what is that?
It is a violent barrage of molecules secreted out of the cell and into the surrounding tissue. We're talking about pro-inflammatory cytokines, chemokines, highly reactive growth factors, and a specific class of tissue-degrading enzymes called matrix metalloproteinases. Okay, let's ground this for the listener. Think about what it actually feels like to age. When you wake up and your joints ache, or you notice your skin losing its elasticity. You aren't just feeling some generic wear and tear like a tire losing its tread. What is this SISP cocktail physically doing to the tissues?
Well, imagine just one single senescent zombie cell sitting in the cartilage of your knee. It is actively pumping out immune signaling cytokines. Those signals act like a flare gun, recruiting immune cells to the joint.
But there's no infection there.
Right. The immune cells arrive, find no infection, but the alarms are still blaring. So they cause chronic, low-grade sterile inflammation.
So the joint is constantly swollen and angry for literally no reason.
Yes. And meanwhile, those matrix metalloproteinases, the degrading enzymes, they're literally chewing up the extracellular matrix. They're dissolving the collagen and the elastin that gives your cartilage its shock absorption and your skin its bounce.
So one microscopic zombie cell is actively rotting the healthy tissue around it.
It gets worse. The SASP signals are so potent that they can actually induce senescence in neighboring, perfectly healthy cells. It is literally contagious.
Wow.
This is why a tissue can rapidly deteriorate once a certain threshold of senescent cells is reached.
It's like a localized biological fire.
And as we chronologically age, these senescent cells accumulate everywhere. They build up in our liver, our heart, our blood vessels, our brain.
Just everywhere.
The systemic accumulation of these toxic cells is now recognized by modern science as a primary driver of atherosclerosis, osteoarthritis, neurodegeneration, and immunosenescence, the general collapse of the immune system in the elderly.
So aging isn't just the passive passing of time. It is an active, aggressive biological program driven by a structural failure at the exact tips of your chromosome.
It is a systemic poisoning from within, triggered by the ticking of the telomere clock.
And what's really striking in the sources is that this clock doesn't just tick at a steady, uncontrollable rate. We actually have an enormous amount of agency here. The way we live our lives physically interacts with this molecular machinery.
It does. We established that the end replication problem, the telomerase falling off, causes a steady baseline of shortening, say 50 base pairs per division. That's unavoidable.
Right.
But that process can be wildly, violently accelerated by environmental and lifestyle factors. And the primary culprit is oxidative stress.
Oxidative stress. We hear that buzzword all the time in health deep dives. That means free radicals, right? The unstable molecules caused by a poor diet, smoking, chronic psychological stress, lack of sleep, pollution.
Precisely. And there is a very specific physical reason why free radicals destroy telomeres faster than the rest of the genome. Remember the sequence of the telomere?
TTAG.
It is incredibly rich in guanine bases. Guanine is a large molecule, and chemically, its electrons are positioned in a way that makes it highly susceptible to oxidation.
It's basically biological rust.
Yes. When you have high levels of reactive oxygen species, free radicals flooding your cell, they aggressively attack the DNA. And because the telomere is basically a giant wall of guanine, it acts like a sponge for oxidative damage.
It takes the hit.
The free radicals physically alter the guanine, turning it into a damaged lesion called 8-oxoguanine.
And how does that affect the shortening?
Well, when the cell tries to divide, the DNA polymerase train comes chugging down the track. But when it hits one of these oxidized guanine lesions, it derails, it stalls out, or it creates single-strand breaks where the DNA simply snaps.
So instead of losing just a little 50 base pair chunk at the very end because of the primer issue, the track is breaking apart much earlier on.
Exactly. A highly oxidatively stressed cell might lose 200, 300, or even 500 base pairs of telomere length in a single cell division.
Wow. So to put that in plain terms, smoking a pack of cigarettes, eating a highly processed inflammatory diet, or enduring chronic psychological stress is quite literally physically fast-forwarding your biological clock. You're snapping off massive chunks of your own shoelace caps.
You are rapidly accelerating your approach to the Hayflick limit. The sources also explicitly note that environmental exposures like heavy metal toxicity, lead, cadmium, can accelerate this shortening by generating highly localized free radicals right next to the DNA backbone.
That is terrifying.
Yeah.
But also incredibly empowering. Because the reverse must be true.
It is. Healthy lifestyle interventions, antioxidant-rich diets, rigorous cardiovascular exercise, deep sleep, stress reduction, they lower your systemic oxidative burden. They sweep up the free radicals.
Now, to be clear, meditating and eating broccoli doesn't turn telomerase on.
Right.
It's not building new telomeres.
Right. It is not magically turning the factory back on in your somatic cells, but it stops the accelerated catastrophic damage. It allows the clock to tick at its natural, inherent, much slower baseline rate. It protects the track you have left.
Okay, so we've mapped out the problem. Short telomeres cause toxic zombie cells, and toxic zombie cells cause aging, tissue rot, and disease. So let's ask the most obvious question in the world.
I think I know exactly where you are going with this.
If the TERT blueprint is sitting right there in my cells, why don't we just find a chemical or a gene therapy to flip the switch, turn the factory back on in every cell in the body, rebuild the shoelace caps, flush out the zombie cells, and let human beings live in a state of youthful homeostasis forever?
It sounds like the most elegant, obvious therapeutic target in the history of medicine, but it brings us face to face with the ultimate biological catch-22. Because while scientists were dreaming about turning telomerase on to cure aging, they looked at the pathology of human disease and realized that nature had already run this exact experiment.
And the result of that experiment is cancer.
The result is the most devastating, intractable disease we face. The clinical data in these sources is unequivocal. Telomerase is inappropriately reactivated in approximately 85% to 90% of all human malignancies.
90%. So it's not just a quirk of one weird subtype of tumor, it is a universal hallmark of cancer.
It is an absolute, non-negotiable requirement for a tumor to become clinically lethal. You have to think about what a cancer cell actually is. It's a cell that has acquired genetic mutations in oncogenes and tumor suppressors that tell it to divide uncontrollably. It's a runaway train.
But even a runaway train is still running on the same track. It's still subject to the end replication problem.
Exactly. A precancerous cell is still subject to the Hayflick limit. If a mutated skin cell starts dividing 50 times faster than it should, its telomeres will shorten 50 times faster.
Right.
Very quickly, it will grind its telomeres down to nothing, the DNA damage response will kick in, and the tumor cell will be forced into senescence or apoptosis.
So the tumor would literally exhaust its own lifespan and die before it could grow larger than the head of a pin.
Precisely. A tumor cannot grow to a lethal size if its cells only have 40 divisions left. It physically cannot generate the billions of cells required to form a mass,
Unless it figures out how to pick the padlock on the TERT gene.
Yes. If a rogue cancer cell can acquire a mutation that forces the telomerase factory back online, it achieves the holy grail: runaway cellular immortality. It can divide infinitely.
And the alarms just never ring.
The telomeres never shorten, the ATM and ATR alarms never ring, and the tumor expands without any structural restraint.
How are these cancer cells actually pulling this off? I mean, how do you pick a genetic lock?
The sources detail a few insidious mechanisms, but the most prominent involves what are called TERT promoter mutations. The promoter is a regulatory region of DNA sitting just upstream of the TERT gene. It acts as the control switch.
Declare the switch.
In incredibly aggressive cancers like melanoma, glioblastoma in the brain, and certain types of thyroid cancer, researchers find highly recurrent specific point mutations right in this promoter region.
A point mutation meaning just a single letter of the genetic code is swapped out.
Yes. One specific letter changes, often referred to as the C228T or C250T mutations. That single tiny change in the genetic code creates a brand new docking station, a binding site, for transcription factors, specifically proteins from the ETS family.
Transcription factors are the proteins that turn genes on.
Right. These ETS proteins naturally float around the nucleus. When they see this newly mutated binding site, they latch onto it, and they forcibly crank the TERT factory back online.
It is a brilliant, terrifying molecular hijacking. The cancer tricks the cell into rebuilding its own immortality engine.
Exactly.
What about the remaining 10 to 15% of cancers, the ones that don't reactivate telomerase? Do their telomeres just keep shortening?
No, they would die if they did. They find a different, even more chaotic workaround. It's a mechanism called ALT, alternative lengthening of telomeres.
How does ALT work without the enzyme?
It relies on a process called homologous recombination. Essentially, the cancer cells use the telomeric DNA from one chromosome as a physical template to copy and extend the telomeres of another chromosome.
They are cannibalizing each other.
They are sharing the wealth. They physically intertwine their chromosomes, often clustering together in massive structures called PML bodies, and they swap genetic material back and forth.
That sounds messy.
It's a sloppy, highly unstable way to maintain length, and it results in massive genomic mutations, but it provides just enough track to keep dividing.
So whether it's by elegantly hijacking the telomerase promoter or by using this chaotic ALT mechanism to steal DNA, a tumor absolutely must solve the telomere problem to survive.
If we connect this to the bigger picture, it explains the fundamental architecture of human aging. Evolution is ruthless. It calculates risk.
What kind of risk?
For a large, long-lived, complex organism like a human being, which requires trillions upon trillions of cell divisions over a lifespan to maintain tissues, the statistical probability of a cell acquiring a cancer-causing mutation is basically 100%.
We are guaranteed to develop precancerous cells. We probably have them in our bodies right now.
Almost certainly. So as a primary defense mechanism, evolutionary biology made a brutal, brilliant calculation. It permanently shut down telomerase in our somatic cells. It instituted the Hayflick limit as an inherent, built-in tumor suppressor mechanism.
It built a kill switch into our biology.
By ensuring our cells have a finite, structurally limited lifespan, the body ensures that most of those rogue, rapidly dividing precancerous cells will hit the telomere wall, trigger the DNA damage response, and die long before they can mature into a fatal tumor.
So our entire lifespan is a tightrope walk. Evolution actively chose to accept cellular senescence, tissue degeneration, arthritis, heart failure, and ultimate mortality as the inevitable, necessary trade-off for protecting us from cancer during our reproductive years.
Yes. Aging is not an accident. Aging is the biological price we pay to not be entirely consumed by tumors by the time we're 30 years old. We're quite literally designed to degrade.
That is an incredibly sobering, heavy realization. But human ingenuity has never been very good at accepting biological limits, right?
No, we really haven't.
Which brings us to the business and clinical reality of all this. Because despite the overwhelming risk of cancer, the anti-aging industry, the translational gerontology market, is booming, and telomeres are the star of the show.
It is a multi-billion-dollar industry built on a single premise. If short telomeres cause aging, we must lengthen them.
Let's look at the actual clinical data from the sources. We have an entire ecosystem of supplements and therapies claiming to hack this system. Let's start with one of the most famous, TA65.
TA65.
This is a botanical activator purified from the astragalus root that claims to safely activate telomerase. What does the rigorous clinical data actually say?
TA65 is a fascinating case study in the enormous gap between moving a molecular biomarker in a petri dish and actually improving human health.
Okay.
The clinical data on TA65 is quite robust, but only within very narrow specific parameters. Studies show that it does, in fact, demonstrate telomerase activating efficacy.
Okay, so it isn't snake oil. It genuinely turns the enzyme on.
It does. It has a measurable effect. And particularly in older adults, randomized controlled trials show that taking TA65 over a sustained period actually elongates critically short telomeres.
Wait, really?
Yeah, you can draw the patient's blood, isolate their white blood cells, run the assays, and physically see that the median telomere length is longer after a year of treatment.
That sounds like a massive breakthrough. I mean, they prove they can wind the biological clock backward.
Molecularly, yes. But clinically, functionally, it is largely a failure.
What do you mean?
The sources point out the critical flaw in the data. While TA65 physically elongates telomeres in the blood, it fails to significantly improve functional aging metrics in the patient.
Wait. Their DNA looks younger under a microscope, but the patient's body doesn't.
Correct. When researchers measure the things that actually matter to a patient's life, cardiovascular function, lung capacity, cognitive vitality, bone density, their overall frailty index, there is no significant systemic reversal of aging.
No reversal at all.
The sources note there might be some modest, localized benefits for the immune system in highly specific older populations, but it does not reverse systemic aging.
How is that possible? If the fraying shoelaces are the root cause of the zombie cells and the zombie cells cause aging, shouldn't fixing the shoelaces fix the aging?
You're touching on what researchers call the single biomarker flaw. Translational gerontology has fallen into a reductionist trap. Aging is not just short telomeres.
It's more complicated.
Telomere attrition is just one of the hallmarks of aging. True aging is a multidimensional, cascading failure of systems. Severe mitochondrial dysfunction, epigenetic degradation where the cell forgets how to read its DNA, loss of protein homeostasis, and the exhaustion of stem cell pools.
So fixing just the telomere length is like putting a fresh coat of paint on a house that has severe termite rot in the foundation and a collapsing roof.
That is a perfect analogy. You made the siding look brand new, you fixed the biomarker, but the house is still collapsing. And furthermore, the sources emphasize a severe theoretical risk that the industry often downplays.
The cancer risk.
Yes. While short-term, small-scale clinical studies haven't explicitly shown TA65 causing massive cancer outbreaks, the long-term carcinogenic potential remains completely unaddressed.
Because you're taking away the kill switch.
If you are systemically taking a pill that activates telomerase across your entire body every day for decades, you are inherently disarming your body's primary tumor suppressor mechanism.
You're taking the padlock off the factory door and hoping the rogue cells don't sneak in.
The clinical consensus in the literature is incredibly cautious. TA65 epitomizes the inadequacy of single biomarker approaches. Until we have decade-scale, massive human trials assessing disability-free survival and tracking cancer incidence rates, these systemic activators remain highly investigational and theoretically dangerous.
So what about other approaches? The sources also dig into a very well-known synthetic compound in the underground longevity community called Epidolon. It has a massive following online, people injecting it, claiming it's a miracle anti-aging peptide.
Epidolon is a synthetic peptide, originally derived from research on natural extracts from the pineal gland. Its massive fame in the longevity space traces back to a foundational, landmark study published in 2003 by Vladimir Kavinson and his team of Russian researchers.
What did that 2003 study actually show?
It reported something extraordinary at the time. They took human somatic fetal fibroblasts, those same connective tissue cells Hayflick used, and exposed them to Epidolon in a Petri dish.
And what happened?
The study showed that Epidolon directly induced telomerase activity, physically elongated the telomeres, and pushed those cells far beyond the Hayflick limit.
It proved that a peptide can grant a degree of cellular immortality in vitro. The cells just kept dividing. That completely explains why the internet is obsessed with it.
Exactly.
But the sources provide a modern clinical evidence scoring for Epidolon. How does it stack up today?
It stacks up terribly. It receives a D grade.
A D.
Specifically, a 31 out of 100 on a rigorous standardized biomarker evidence scale used by clinical gerontologists.
A 31 out of 100. Why so incredibly low if the 2003 data physically proved it works on human cells?
Because in modern clinical science, making cells divide in a plastic dish is just the starting line, not the finish line. Harsh grading comes from a complete and total lack of controlled, long-term human safety data.
We are flying blind regarding the cancer paradox again.
Completely blind. The evidence score explicitly states that because systemic telomerase activation is the foundational hallmark of cancer biology, the theoretical risk of Epidolon triggering, accelerating, or immortalizing undiagnosed microtumors in a human body is massive.
Because we all have microscopic clusters of mutated cells.
Right. And the only thing keeping them in check is the Hayflick limit.
And people are injecting a peptide to bypass that exact limit.
Yes. And without rigorous, double-blind, decade-long human safety trials, which do not exist for Epidolon, that cancer risk is impossible to quantify.
So what does this all mean for you as the consumer? Are these therapies, botanicals, peptides, just molecular parlor tricks?
Mm.
We're lengthening the DNA in a lab or in a specific white blood cell, but the patient isn't functionally getting any younger, and they might be actively fertilizing undetected cancer cells.
For systemic whole-body telomerase activators taken as unregulated oral supplements or unverified peptides, that is the current, sobering reality of the hard science. It is a biological parlor trick that aggressively ignores the staggering complexity of human aging and oncogenesis.
But the rigorous scientific community hasn't given up on telomeres. This takes us to the truly cutting-edge research. If systemic permanent activation via pills is dangerous and ineffective, how are the real researchers trying to thread this needle?
They are abandoning the idea of taking a daily pill, and they are turning to highly targeted viral gene therapy.
Gene therapy.
The sources detail a remarkably innovative strategy pioneered by researchers like Maria Blasco. They are using AAV adeno-associated viruses to deliver the TERT gene directly to specific tissues in living animals.
Using a virus to infect a cell with the telomerase blueprint. Okay, let's unpack this. How does using a virus solve the cancer problem? Doesn't that just turn the factory on forever?
The brilliance of this approach lies entirely in the specific vector they chose. Adeno-associated viruses are engineered to deliver genetic material, but critically, AAV vectors are non-integrating.
Non-integrating.
Right.
Meaning they don't splice the new DNA permanently into the host cell's chromosomes.
Exactly. When the AAV delivers the TERT gene into the nucleus, it doesn't stitch it into the genome. It exists as an episome.
An episome.
It's a temporary, free-floating piece of circular DNA inside the nucleus.
Why does that matter?
Because it provides transient expression. The cell reads that free-floating episome, transcribes the telomerase, and starts rapidly rebuilding the telomeres. Works beautifully. But as the cell divides and multiplies, that non-integrated viral DNA isn't copied along with the rest of the chromosomes.
So the virus gets diluted.
It gets progressively diluted out over a few cell divisions. Eventually, after a few weeks, the episome disappears completely, the TERT gene is gone, and the factory shuts down again.
That is absolute genius. It's a molecular hit-and-run operation.
It really is.
The telomerase comes in, does a massive burst of repair work to rebuild the shoelace caps, heals the tissue, and then vanishes completely before any potential precancerous cell can permanently hijack it.
You get all of the powerful, regenerative, telomere reparative actions while radically minimizing the long-term oncogenic risk. And the preclinical data in animal models is staggering.
What do they find?
Researchers demonstrated that if you induce a severe myocardial infarction, a massive heart attack in mice, and then deliver this AAV TERT gene therapy directly to the heart, the tissue regenerates.
The heart heals itself.
Yes. The heart tissue, which normally cannot regenerate and just forms a dead scar, actively repairs itself. Furthermore, when they applied this gene therapy systemically to normal, healthy adult mice and even very old mice, it significantly delayed the onset of age-related pathologies.
And did they live longer?
It extended their median lifespan by up to 40%.
40%. That is a massive extension of life. And did those mice all die of cancer?
That is the most important data point. No. It extended their lifespan by 40% without increasing cancer incidence compared to the control group.
That is incredible.
It proved in a mammalian model that if you can strictly control the dose, the location, and the duration of telomerase activity, it has profound, genuine anti-aging effects. The challenge now is scaling that complex viral delivery system to safely work in a human body.
Okay, so that's the futuristic anti-aging side. But let's completely flip the script. We've established that 90% of human cancers rely on telomerase to survive. So why are we spending all this time trying to give telomerase to people?
Yeah.
Why aren't we ruthlessly attacking it to cure cancer?
We are. And this is where the telomere field has transitioned from experimental biology into a major FDA-approved clinical success.
Oh, wow.
The oncology field recognized decades ago that telomerase is the ultimate Achilles heel of cancer. If you can develop a drug that successfully blocks telomerase, you could theoretically starve almost any tumor to death, regardless of what mutations caused it in the first place.
Just cut the fuel line to the immortality engine.
Precisely. And this decades-long effort culminated recently with the 2024 FDA approval of a highly anticipated drug called imetelstat, marketed under the brand name Rytelo.
Let's get into the mechanism of Rytelo. What exactly is imetelstat, and how does it shut down a factory that is locked inside the nucleus?
It is a marvel of targeted drug design. Imetelstat is an oligonucleotide telomerase inhibitor. Remember, TERK.
Yes, the RNA template blueprint that telomerase requires to know what sequence to build. The instruction manual with the TTAG code.
Imetelstat is a synthetic, heavily modified lipid-conjugated piece of genetic material. It is designed by scientists to be the exact, perfect, complementary mirror image of that TERK template.
So it's designed to stick to the blueprint like Velcro.
Yes. It acts as a highly potent competitive inhibitor. It physically infiltrates the nucleus, finds the assembled telomerase complex, and locks directly onto the active site of the RNA template.
And then it just won't let go.
It binds so tightly that the TERT engine cannot read the blueprint.
It jams the gears in the factory.
It completely shuts down the assembly line. The cancer cell can produce all the TERT protein it wants, but it's totally useless.
That is an incredibly elegant molecular assassination. But let's look at the clinical application. Is this approved as a general cure for all cancers? Can I take it for melanoma or lung cancer?
No, the initial approval is highly specific. We are looking at the actual FDA prescribing information from the sources. And Rytelo is currently indicated for adult patients with low to intermediate 1 risk myelodysplastic syndromes, or MDS.
MDS. Let's break that down for the listener. That is a type of blood cancer, right? It originates in the bone marrow.
Correct. In MDS, the blood-forming stem cells in the bone marrow become malignant. They mutate, they turn on telomerase, and they begin dividing rapidly, crowding out the healthy stem cells.
So the bone marrow just stops working.
Right. As a result, the bone marrow fails to produce healthy, mature red blood cells, white blood cells, and platelets.
And the specific indication says it's for patients with transfusion-dependent anemia.
Yes, patients whose bone marrow is so compromised that they require constant blood transfusions just to survive, and who haven't responded to other stimulating agents. By administering imetelstat, you forcefully inhibit telomerase within these rapidly dividing malignant bone marrow clones.
And because they are dividing so fast, they burn through their remaining telomeres almost immediately.
Exactly. You force the cancer cells to undergo rapid telomere attrition, driving them violently into senescence and apoptosis. You selectively kill the malignant clones.
That sounds like a massive victory. It proves the concept works in humans.
Right.
But reading through the FDA prescribing info in these sources, the warnings are intense. The side effects are brutal. This is not a gentle, targeted therapy.
Yeah, it is not gentle at all. It illustrates the heavy, terrifying toll of systemic telomerase inhibition. Remember earlier in our deep dive, we discussed that while the vast majority of somatic cells don't use telomerase, there are critical exceptions.
All right. Certain stem cells absolutely require it. The intestinal stem cells, the immune system, and the hematopoietic, the blood-forming stem cells in the bone marrow.
And that is exactly where the toxicity of this drug hits hardest. When you inject a systemic telomerase inhibitor, it doesn't just jam the gears in the cancer cells, it jams the gears in your healthy, life-sustaining stem cells, too.
The administration protocol listed here is intense. It's given intravenously, 7.1 milligrams per kilogram, over a 2-hour infusion every 4 weeks. And the black box level warnings are severe. The primary adverse reactions are grade 3 and grade 4 thrombocytopenia and neutropenia.
Let's translate what that actually means for a patient sitting in a hospital bed. Thrombocytopenia is a catastrophic drop in blood platelets.
And platelets are what allow your blood to clot.
Right. When you hit grade 3 or 4, patients are at risk for spontaneous, severe, potentially fatal hemorrhage. They can bleed out internally.
And grade 4 neutropenia.
Neutrophils are the frontline white blood cells that fight off bacterial infections. A patient with grade 4 neutropenia has essentially zero functioning immune system. A common cold, a minor scrape, or normal bacteria in the gut can rapidly escalate into fatal sepsis.
So, by attacking telomerase to kill the blood cancer, you are simultaneously poisoning the healthy bone marrow stem cells that require telomerase to survive and produce healthy blood and immune cells.
Exactly. It is collateral damage of the highest biological order. The clinical reality of using Rytelo is that you are actively attacking the very regenerative capacity of the patient's bone marrow.
That has to be a nightmare to manage.
Clinicians have to walk an incredibly dangerous tightrope. They have to monitor blood counts weekly. They constantly have to delay doses or reduce the dosage just to keep the patient from bleeding to death or dying of infection while desperately trying to maintain enough inhibition to kill the cancer.
It works, it saves lives, but the therapeutic window is incredibly narrow. It really emphasizes the core theme of this entire discussion. Telomerase is a master regulator of life and death for the cell. You cannot just flip it on with a supplement or flip it off with an inhibitor without shaking the entire foundation of human biology.
It is deeply embedded into the architecture of survival.
Hmm.
Which brings us to the final and frankly perhaps the most surprising revelation in all of the source material today.
I love a good scientific plot twist. After diving through DNA replication, immortal zombie cells, viral gene therapies, and bone marrow toxicity, what could possibly be more surprising than what we've already covered?
Well, consider this. Everything we have discussed for the past 50 minutes has operated on one single, foundational, unquestioned assumption. The assumption that the only job of the TERT protein is to sit inside the nucleus, bind to the TERT RNA blueprint, and build TTG telomere DNA at the ends of chromosomes.
Right. It's a reverse transcriptase. It builds the shoelace cap. That is literally its name. Telomerase reverse transcriptase. What else could it possibly do?
It turns out that is only part of the story. The sources highlight a massive, ongoing paradigm shift in the field of cellular biology. Decades of dogma are being rewritten.
Rewritten how?
Researchers have discovered that the TERT protein doesn't just hang out in the nucleus. Under specific conditions, particularly conditions of cellular stress, a significant portion of the TERT protein physically exits the nucleus and migrates to the mitochondria.
Wait. The mitochondria,
The
powerhouse of the
that is
cell, the energy factory. What on earth is a DNA-building enzyme doing in the energy factory? There are no telomeres in mitochondria.
Exactly. That is why researchers were so utterly baffled when they first observed it. Mitochondrial DNA is entirely different from nuclear DNA.
How is it?
It is a circular loop inherited entirely from your mother. It doesn't have ends. It doesn't suffer from the end replication problem. It does not need and does not have telomeres.
So why is the telomere building machine going there?
Because we now know that TERT performs entirely different extratelomeric functions when it enters the mitochondria, functions that have absolutely nothing to do with lengthening chromosomes.
What kind of functions? What is it doing in there?
It acts as a profound, localized protector of the mitochondrial machinery. The mitochondria are the primary source of oxidative stress in the cell. When a cell experiences massive trauma like during a heart attack or a stroke, when oxygen is cut off and then suddenly restored, a flood of free radicals is generated inside the mitochondria.
A localized explosion of oxidative stress.
Yes. Normally, this explosion ruptures the mitochondrial membranes. The mitochondria spill their toxic contents into the cell, which immediately triggers the apoptosis cascade.
The cell rapidly kills itself.
Right. But researchers discovered that if the TERT protein is present inside the mitochondria during that stress event, everything changes.
How does TERT stop it?
TERT binds to mitochondrial RNA, not telomere DNA. It physically interacts with the mitochondrial genome to reduce the production of those reactive oxygen species.
Wow.
It stabilizes the mitochondrial DNA, enhances energy output, and physically blocks the signals that trigger apoptosis.
It acts like a blast shield for the power plant.
It maintains mitochondrial integrity under extreme lethal stress, completely independent of its ability to lengthen nuclear DNA. It is a dual-function protein. It is an architect in the nucleus and a firefighter in the mitochondria.
This raises an incredibly important question.
Yeah.
And it kind of turns everything we talked about in the longevity segment upside down.
Oh, so?
If the TERT protein has these incredible protective, anti-aging, energy-preserving effects in the mitochondria that have absolutely nothing to do with lengthening chromosomes, does that mean the entire longevity industry has been staring at the wrong metric?
You've hit on the bleeding edge of the science. For 30 years, we have been obsessed with telomere length as the sole metric of biological age.
Right, because of the Hayflick limit.
We thought the magic was in the length of the track. But what if the true key to cellular longevity isn't the physical length of the DNA at all? What if the real magic is the overall metabolic and mitochondrial health governed by this extratelomeric role of the TERT protein?
That would perfectly explain why TA65 lengthened telomeres in the blood but didn't make people functionally younger. Maybe it was just forcing the nucleus to build DNA, but it wasn't helping the mitochondria produce energy.
Precisely. It was treating the symptom, not the underlying metabolic decay. And this revelation is driving the absolute cutting edge of experimental therapeutics today.
Like what?
The sources mention entirely new therapeutic platforms being developed right now, like therapies utilizing a highly modified version of the protein called catalytically inactive HTERT.
Catalytically inactive, meaning they engineer a version of the TERT engine that is deliberately broken. It can't build DNA.
Yes. They mutate specific amino acids in the active site. They engineer a version of the TERT protein that has zero reverse transcriptase activity. It is physically incapable of adding T-TAG to the chromosomes.
So it can't push a cell past the Hayflick limit?
It cannot grant runaway cellular immortality to a tumor.
It completely sidesteps the entire cancer paradox.
It theoretically eliminates the massive oncogenic risk that has plagued this field from day one.
But if it doesn't build telomeres and it doesn't lengthen the DNA, what good is giving it to a patient?
Because it retains its other, arguably more important functions. Even though the engine is broken, the structural presence of the TERT protein can still physically help cap the ends of the chromosomes, silencing those chronic ATM and ATR DNA damage signals that create the toxic senescent zombie cells.
Oh, wow.
And crucially, this catalytically inactive TERT can still migrate to the mitochondria.
It can still act as the firefighter.
Yes. It enters the mitochondria to restore metabolic function, neutralize the oxidative stress, and heal degenerating tissues by rescuing the cell's energy grid.
So they're actively trying to decouple the genuine anti-aging metabolic benefits of the TERT protein from the cancer-causing risks of physically lengthening the DNA. That is brilliant.
It is a total reconceptualization of the biology of aging. It suggests that reversing cellular decline isn't about giving the cell an infinitely long shoelace to trip over.
Right.
It's about calming the false alarms in the nucleus and protecting the breathing, burning energy grid in the mitochondria.
Okay, we have covered a massive amount of incredibly complex ground today. Let's try to synthesize this journey. We started by looking at the physical reality of the biological clock.
The telomeres.
Right, those millions of repeating DNA letters acting as the aglets on our genetic shoelaces, shielded by the intricate knot-tying folds of the shelter and witness protection program.
And we saw how the fundamental mechanical glitch of DNA replication, the end replication problem, acts as the unyielding ticking mechanism.
It slowly, inevitably frays those caps until the cell hits the Hayflick limit, undergoes that massive epigenetic shift, and becomes a toxic, inflammatory zombie cell that pumps out the SSP, driving the physical decay we call aging.
Then we looked at the magnificent machinery of telomerase, the TERT engine and the TERT blueprint, the ultimate double-edged sword.
It is the fountain of youth that our own biology deliberately locks away to protect us from cancer, only to be ruthlessly hijacked by 90% of tumors to achieve a terrifying, lethal immortality.
And we examined the harsh clinical reality, the failed promise of single biomarker supplements that can lengthen DNA without actually improving human health, the dangerous, unquantifiable unknowns of untested peptides.
The brilliant but complex hit-and-run tactics of non-integrating viral gene therapy, and the brutal bone marrow-crushing reality of targeting telomerase in oncology with drugs like imetelstat.
It truly is a masterpiece of biological complexity.
Which brings us to a final, lingering thought for you to take away from this deep dive. We've spent decades obsessively staring at the ends of our chromosomes, measuring the physical ticking of the clock in base pairs, holding on to this belief that if we could just find a way to add more sand to the top of the hourglass, we could cheat death.
We were so focused on the length.
But if the active engine of telomerase, TERT, has this secret, profound second life, migrating away from the DNA entirely to protect our mitochondria, regulate our oxidative stress, and maintain our metabolic energy completely independent of telomere length. What if the true secret to reversing cellular aging isn't found at the fraying ends of our chromosomes at all?
It makes you wonder.
What if the secret to youth is actually found deep in the energetic, breathing heart of the cell?
It makes you wonder what other profound secrets our biology is hiding in plain sight, just waiting for us to look past the dogma.
Exactly. Thank you for joining us on this deep dive into the biological ticking clock. Keep questioning the world around you, keep pushing past the simple answers, and we'll catch you on the next one.